Okay, Anna.
Okay.
Okay, thank you, everybody. Greetings to all from Nairobi, Kenya. My name is Ana Oliveira. I'm the BR, Breeding Resources Genotyping Coordinator from CIMIT, based in Nairobi. And as we convene for the second seminar in the series Crop to End Hunger, Case Studies in Africa and Beyond, supporting the CGIAR partners through genotyping services, I extend warm regards to each participant from their respective locations. So in our inaugural seminar on August 10th, Mokhtar Kante and Guilherme Silva, they provide some insights into the utilization of KASP markers in potato and sweet potato. So today's seminar features Dr. Agre Paterne. He's a molecular geneticist at IITA, who will delve into the application of genotyping in yam. Following his presentation, we will allocate some time for some questions and answers, and where we will have some interactions. But before we start today's presentations, we are very honored to welcome Dr. Robert Azevedo. He is the Emeritus West Africa Hub Director at IITA Nigeria. Dr. Robert, your gracious acceptance of this invitation is highly appreciated. Thank you very much. And without further ado, the platform is yours. Dr. Robert, please.
Thank you very much, Anna. Hope you can hear me well. Yeah,
Yes.
there are exciting developments in the genetic improvement of yams in West Africa in particular. And this is led by IITA, but there is a very active collaboration with national programs in 4 countries in particular: Benin, Cote d'Ivoire, Ghana, Nigeria. But there are also collaborative efforts in this that has been started in Democratic Republic of Congo and Ethiopia. And the developments have been aided by improving understanding of the genetics and genomics of the crop, building on the past efforts, including the publication of the reference genome. And you hear a bit more about this in the presentation that will be made. There is increasing efficiency, including through the development and use of more precise tools. And this has been aided by interactions with the Excellence in Breeding platform and associated projects like the Crops to End Hunger, interaction with the Polyploids group, and partnership with Bayer through the Modern Breeding Project, and also in particular the sustained support that we have received from the Bill and Melinda Gates Foundation for Yam Breeding in West Africa, which in addition to the national programs, linked us up with Cornell University, CIRAD, and the James Hutton Institute. I should also mention the relevant collaboration we've had with partners in Japan, in particular the Iwate Biotechnology Research Center and JIRCAS. While this is going on, it's important to note that there has been corresponding improvements in the phenotyping tools and the capacities of the national programs and seed systems, which are important for the delivery and scaling of new varieties. And I need to acknowledge that the sustained support for development of the seed system has also come from the Bill and Melinda Gates Foundation. Asrat Amele, the senior breeder, has been leading the team in West Africa and extending to other partners elsewhere also. And he's been strongly supported by Agre Paterne, the molecular geneticist. And their work is characterized by effective collaboration with colleagues in other disciplines, especially in developing the tools for phenotyping, for instance, and also close interaction with the NAS breeders, including the joint release of new varieties, degree-related training of graduate students, and regular publication of their results. Agre joined the team in 2016 and initially focused mainly on analysis of molecular data And he benefited from training at the Cornell University in bioinformatics, quantitative genetics, and data analysis. And his responsibilities included data curation, managing the YAM base, where the genotyping data and other data from the YAM programs are deposited. He has been a molecular breeder in the team since 2019. And from this role, he's been contributing to the achievement of the goals of the CGIAR. So Agre will now share with us some of the progress being made in harnessing the potential of highly informative KASP and mid-density SNP markers panel for YAM breeding. So over to you, Agre. Thank you.
Okay. Thanks so much, Doc, for the welcome introduction. And also thank you for inviting us to share our works through this platform. We really appreciate. So I will start the presentation by just saying that we'll just be presenting our work on addressing the potential of highly informative KASP and middle density SNP markers panel for YAM breeding. So why are we using the kind of just my presentation? We try to follow an outline. I will just present a brief introduction on why are we using the marker, and then our results so far on low density SNP markers, how we select the PC marker, and then how we validate them, and then we are using them in the breeding program. Also, we will talk on the trait marker validation, where we are, and then what we intend to do also. After that one also, I will present our result on the middle density SNP markers, the result we have, and then where are we deploy them in the breeding program. I will also share information on the development of reference genome supported by the crop to hunger in collaboration with Corteva. And also I will present the ongoing molecular activities and then the take-home message. So why are we using the middle and low density SNP markers in the breeding program? Generally, is to address some mislabeling error in the breeding program, especially for some multiplication, and also some material that we have them across many locations, just to be sure that we have the true to type genotype over here in generation. Also, we need marker to assure our genetic security and then variety tracking, and then followed by pedigree verification, especially after our crossing, to be sure that what we have, they are true to type, and then they are hybrid of the selected parent. We also need marker for trait prediction, especially at early generation, because if we take the YAM through the classical breeding, it's too lengthy. So with marker, we can try to reduce the breeding cycle. And then this one also can help us to optimize our breeding program. We are also using the marker for parental selection, especially to develop some iterative group, what we are using the breeding program for advancing our material. And also we need some middle density SNP markers for genomic prediction, and also to establish a genomic prediction and cross performance. So the next, how we select our low density SNP markers, especially for quality control and quality assurance in the breeding program. The first activity we did was from whole genome reference with our collaborator IBRC in Japan. So we got more than 6 million SNP markers, but we cannot use all those 6 million SNP markers for quality control. So we put some criteria in place to be able to select most informative SNP markers. So we focus on polymorphic information content, the ratio of observed and expected heterozygosity. Then we selected some marker with high minor allele frequency. And then for YAM, it's very important we look at the putative paralog, because the YAM, we have seen that we may be in presence of polyploid, which is the case here. because after implementing this one, we realized that the paralog is above 1.5, which means we are in presence of a polyploid for crops. So that means we need to take that information in content when we want to select those marker information. The GC content also is very important when we want to select some markers. So in our case, the GC content is less than 50%, which means any marker we want to select has to have those criteria. And then the missing information should be very less. And then we remove also the indels. So those are the different criteria we use to be able to select very highly informative SNPs markers. And then after filtering the marker now in the breeding program, we can we now say how many minimum SNP markers do we need in the breeding program to be able to make a very good prediction, irrespective of the activity we want to do. And then we established two methods. One is based on purity, and then the second method is simulated annealing. And then using the two methods, you can see from here that both of them predicting like from 50 to 100 SNP markers, we can predict information up to 100% of what we have. And then using this method now, we now select 50 SNP markers for each of the methods. So that means we are expecting to have 100 SNP markers using the two methods, both the purity and then the simulated method. This process has been developed, and then all the information is available. It can be you can get it from here, which is a GitHub account, so anyone can make use of it. So moving forward now, using these two techniques, the next thing we did was to just fetch out with the Flunken sequence and then develop a database of the 99 SNP markers. Because rather than 100, one of the SNP markers were identified by the two methods, so we end up with 99 SNP markers. Then to be sure that this marker can be used to predict or can be used for variety identification and species identification in yam breeding, we selected 4 plates which have a different yam species. We have Alata, almost 6 different yam species: Alata, Rotundata, Quincylis, Esculenta, and Kinesis, which was sent to Intertech and with the support of EIB also. And then the first result we got, you can see for the 4 plates we sent here, 99 SNP marker tested, we have a color rate of 63% across all the SNP markers, all the sample we sent. And then in general, you can see for the SNP view, we got a very good information, although some of the markers are not good also. So we will explain how we discard them and then how we are making use of the most informative SNP markers in the breeding program. Moving forward now, from the 99 SNP markers that we validated with the Intertech, we now narrow it down to 50. So these all the results we are going to present now is based on 50 SNP markers. And then using the 50 SNP marker, and then across all the 6 different yam species, you can see that we have all the Rotundata group in one place, which means the marker is very effective in identifying first the yam species. In this group, you have a cluster of Quincylis with no mixture here, and then you have a cluster of the Kinesis here with no mixture. And then we have a cluster of Esculenta, Alata, and then Bobifera, which has been previously reported in a lot of publications using a million of SNP markers. So using firstly 50 SNP marker, we can identify species. Then we move forward also, and then during the validation process, we intentionally use some duplicates, some sample that we know that they are duplicates, or we replicate them in the breeding program just to see if the marker will be able also to predict them as duplicate. And then you can see some of those samples here, which we intentional duplicate also appear that the marker also identify them as duplicates. So which means we can use these markers to identify duplicate in the breeding program as well as to identify yam species. The same approach also was, we did the same thing also using principal component analysis, and then you can see we have a group of Rotnatafia, and then we have our Princilis, and then kinesis, and then a mixture of Alata, as we have seen through the phylogeny analysis here. And then we make use also the same marker just for the population structure analysis, and then we can see the Alatafia, Princilis, which means with this set of markers, we can use this marker for a lot of activity in the breeding program. So moving forward, what we did again in 2020, we have used a polycross in the breeding program, whereby we established a female plant surrounded by some male, and then we let a natural pollination happen. And then we use a 6,000 SNP marker to do pedigree reconstruction, which was published in 2020. And then after we validated this recent QC marker, what we did, we selected the 50 SNP marker again and made use of this one also to reconstruct the same pedigree information, which means also we can use this 50 SNP marker now to predict for parental analysis in the breeding program if we have a cross. So those markers, they are very useful in the breeding program. And then recently, we are also trying to develop some interspecific variety in the yam breeding program from savanna adapted, which D. abyssinica, and then from the rainforest, we have the D. princilis. And then we make a crossing between these two species, and then from this one, we got a progeny. So from the progeny here, you can see this as our hybrid we got after making the crossing. And then what we did now, we now selected the same markers, and we validate them, and then we use them to see if we have some hybrid in the interspecific. Looking at the two parents here, what we did also, we divided the markers segregation for the two parents, the homozygous and then the heterozygous. So we look at the homozygous markers for the two parents, and then also we focus now on the progeny. And then looking here now, you can see some of those one did not meet what we are expecting based on the two, based on the information of the parent here. So this one, firstly, we are consider them as to be non-hybrid, but we want to support this information using a high density. So we have sequenced this one with Illumina with ongoing projects. So we compare the results with what we have to be able to make a very the final decision. And then last year also, we have a lot of material, especially our checks that we use across several locations, from almost more than 10 locations. And then we wanted to be sure that our checks are the same material across different locations. And then what happens here, if you look at probably some of these material here, which is one of our checks, or 2665 is also very common in the breeding program. You can see that across some of the locations we are seeing some level of mixture, which means this marker can help the breeding program for kind of like genetic purity, to be sure that what our material we are using over generation for one location to another one is the same thing, to avoid any misleading information in the breeding program. The same thing also for one of our popular variety, which is Oyuyao. The same thing also was done, and then we can see how those markers are performing. Also, how those markers identify them across location. The next activity also we did under the low density SNP markers is for the trait marker, the trait validation. And then for this one, we have identified a lot of SNP markers through the association mapping as well as the linkage mapping. But all those markers we have identified, some of them are not really informative. So we kind of like establish approaches to identify promising markers, and then we validated some. These are the current traits we have identified, some SNP markers associated, and then we have the source. This can be through association mapping, and then also some of coming from linkage mapping. This is the number of QTL we identified, and then the promising SNP QTL we identify, and then this is the number of marker we validated. So how we proceed to identify the promising SNPs, the promising QTL, and then how we validate, that is the next, my next slide. So to select the promising marker, what we did, we just picked the case of yam mosaic virus. And then the first thing we did, we developed haplotype segregation in the breeding program, especially for the marker we have. And then if you look at this marker that we identified to be associated with yam mosaic virus, you can see for some of those haplotypes, the 3 haplotypes, this one there could not be used to discriminate the population based on the disease information. And then if we look at this one also, we have, this is very useful information because with this marker, you can use this one to predict based on the haplotype information. So using this one and then the marker prediction effect, we're able now to know which one is the favorable alleles which are associated to the disease tolerance, and then which one are associated to the disease susceptible. For the case of this marker, for example, we know that the low disease is what we are looking for, and then you can see the J is associated with low disease, while the A is associated with the susceptible. So which means the presence of A increase the disease, while the J is responsive to predict the low disease. So this is the way we proceed to identify the promising SNP markers in the breeding program. The second approach we also use to confirm or to identify, to select the promising SNP markers is how close the gene is, the target gene is to the marker we are looking for. So we establish a lot of information based on the SNP marker position and also based on the interval where we identify the gene. And then from there now, we can easily see here now how closely the gene is. If the gene is close to the marker, we focus on those ones. But if it's very far to the marker information, we will not be able to consider that marker for validation and then to be advanced. That now focusing on all those information now for in the breeding program, we have validated the marker for 7 different traits, and then we are yet to deploy them in the breeding program for marker assistance selection. So what we did for the validation for the trait like sex, we selected some genotype with well-known sex. The sex is known, while we pick some individual that we don't know the sex, and then we proceed for sequencing, and then we test those marker on them. The same approach also was adopted for the disease, also susceptible, and then tolerant to disease also. And that's what we did. And then for the plant cells, now we make use of bootstrapping through the regression analysis, and then we can see for some of those markers for the plant cells, we have a prediction accuracy of 85% and then a 92%. And then, as you can see here now, the homozygotes is associated to the male information, while the heterozygotes is associated with the female, which is kind of like very good information, as this one aligns very well with what our collaborator in Japan has reported. The only information we are working in collaboration with our collaborator from Japan is to see if we can have more information about the marker to predict for some genotype that generally they don't give a flower. So we hope to improve the marker so that at early generation we'll be able to give a very clear prediction to the breeder to assist them in advancing the product. We also make use of this for the flowering intensity, and then also we have the favorable alleles, and then for the flower intensity, we are looking also for the A, which is kind of like predicting for high flower, while the G is associated with low flower intensity. And then for this one, for the 4 marker we work on, we have a 2 to 3 that are very good because the prediction accuracy is above 60, while for this one is not really good. We have 31% prediction accuracy. The same thing also for this one, whereby we have 0.76% for accuracy for the flower intensity, and then for the 4th marker we have 0.96 for the flower intensity, and then you can see that this marker can really identify high intensity both for male and female, which is very useful for the breeding program. For the yam mosaic viral also, we did the same thing, and then in the breeding program, we have a marker that's kind of like we're predicting for susceptible and then for tolerance. And then here now we have this marker that is associated with a low disease information, which is associated to the J, is the favorable alleles, what we are looking for. And then we have also the T and then the J, especially the T is the unfavorable alleles, which is associated with the susceptible. So currently for the yam mosaic virus, we have very good markers that we are intending to use in the breeding program. I hope by this year we'll be able to deploy them in the breeding program. And then we are already in discussion with Intertech to initiate this process. But for the yield, the marker we have is not really good because the yield is kind of like a complex threat. We have a 0.31% prediction accuracy, and then we have 0.54% prediction accuracy. This one also, we are working with the crop lead, which is Dr. Asrat, by using historical data more than 11 years to see how best we can identify additional SNP markers using some other information, either additive and dominance, to combine with what we have to be able to come out with a marker for the yield, what we can use that, especially at the early generation to establish a very good genomics selection approaches. The next activity we did, and then this one is still in process for the final validation. We are still in discussion with Crop2, Enga, Anna, and Eng. I think they are in loop on this one. We recently identified a middle density SNP marker, which is around the 3,000 SNP markers. So the initial SNP marker is how it looks. I think here we have more than 200,000 SNP markers. And then we look at the LGBK, how this one is well distributed on the genome, and then where we have the first drop. And then after making a lot of criteria and then discussion, we came up with 392 SNP markers. So the 392 SNP markers, they were also well distributed across the genome. And then what we did also here, we tried to include the QC marker that we presented as alongside with the traits markers. So these 3,000 SNP marker that we have here, at the same time that we are using for middle density, especially to target the genomic prediction, we also have information on the trait associated marker. So anytime we use this marker, we can have some additional marker on the trait that carry the alleles of interest in the breeding program. So this one, we have validated the sequence, so we are just here to send the sample to Intertech for final development and then deployment in the breeding program. So moving forward now with the help of Crop2, Hunger, and then Corteva and CIMMYT, we are able to develop additional 4 reference genome to accelerate the discovery in the yam breeding program. So the 4 genotype included 2 alata, which is one is female and one is male. And then we also have 2 rotundata, which is the white yam. One is male and then one is female. we are interested in this guagua because it's a semi-dwarf, and then we think that if we can have some very good gene information, it will really be useful in the breeding program, because the cost of staking in the yam breeding is very expensive. So if we could have a product that can be semi-dwarf and then give a reasonable yield without staking, it will be a very good achievement for the breeding program. Ongoing molecular activity in the breeding program, we have identified a lot of SNP markers, especially those that are associated with some key traits. So we now want to finally move to the marker-assisted selection, want to deploy them in the breeding program for the breeding to have a tool to make the selection based on what the marker we predict. We also doing some annotation with some vendor and also some gene model prediction to be able to have a very good gene informative in the breeding program, which will help us to accelerate our discovery. And then, as I was saying before, we are doing some gene expression analysis for semi-dwarf genotype. We hope to get a very useful information to impact the breeding program. So we will validate also the middle density SNP markers, and this one is still an ongoing discussion. So we'll be using this one for product advancement, especially and also for a genomic prediction of cross performance, just to predict the cross merit information in the breeding program. We also have those interspecific generated between the kinases and between the Abyssinica and then Princilis. The progeny, we have tested the marker on them, but we want to combine this information with a quantitative method such as the ELISA method. We'll be able now to, if we identify a very good product from the interspecific, this will be very useful also. We are also doing some variety tracking. We start very soon, especially to monitor our release variety in the farmer field to see the rate of adoption of our release variety using a marker information. And then we implement a genomic selection and prediction very soon. And then currently also we are looking at the effect of heterozygosity level on trait association. This one is very important because the level of heterozygosity in yam is very high. So we are targeting on some genome, some region of the genome which are kind of like highly heterozygous, and see how this one will impact the development of trait association markers. So these are the current molecular activity that is ongoing. Then this is the take-home message. So we have developed in the breeding program, the QC and the quality control and quality assurance marker. And then we are making use of them in the breeding program for routine activities such as pedigree and then variety, genetic purity. We have also developed some trait length markers, and then we have developed them, and then we soon deploy them in the program. And then we are also evaluating some populations to identify more trait marker, especially to the food quality, to have some marker associated to the tuber quality. The middle density SNP marker, we have developed them, and then we are in the process of validating them to be used for product advancement in the breeding program. So we have added we have also developed some additional reference genome. This one with the crop to hen hunger. So this is very useful information. And then to finalize this one, we will continue to make use of SNP markers in the breeding program from the low to the high density, and then also the trait marker we have in the breeding program. We will use them in the breeding to accelerate our product. So my last slide is to thank Excellence in Breeding Program for the different support we have received for them, and especially for some genomic prediction implementation in the breeding program. Corteva, which has supported also for our works, especially for the genomic reference genome. And then the crop to Hanga, Anna and Hank, thanks so much for your support to the yam breeding. The polyploid is very useful, and then we have used a lot of tools developed by the team in the breeding program. And then Cornell, which is really supporting the yam breeding and then hosting most of our genomic information, IT Bioscience to support our genotyping activities. Intertech, I hope Petra is there. Thanks so much for supporting our works. And then the Smith, who hosts the 4 reference genome and then the database on the website. And then the University of Illinois to support for bioinformatics support. With this, we say thanks so much. And then this is my talk.
Thank you, Agre. Thank you very much for this insightful presentation. We have some questions already, and I do invite the authors of the question to unmute and to ask themselves the question to Agre. And the first is Dr. Sara. Can you unmute yourself and ask to Agre the question, please? Okay, so maybe I can ask the question, and if Sarah want to add something. So Agre, what is the role of Corteva in the yam whole genome sequencing?
Okay, and thanks so much. Corteva was responsible to sequence the material. So we ship the material to them. So they were the one that were responsible for the whole genome sequencing information, which was later transferred to Smith.
Sarah, unfortunately, she cannot connect the audio. If there is anything that maybe is not so well said, please just write and we can ask. So the next question is from Dr. Juan Burgueño from CIMIT. Juan, do you want to open the microphone and ask the question?
Yes, thanks, Anna. Thanks for a good presentation, very interesting. My point is, we have found in CIMIT for wheat at least that, and also in maize, that around 20–30% of marker data has technical error. That is because the process of genotyping, what we did really was check the offspring with the parents. We found some mistake on this. Are you considering this rate of error when you use marker data for QC and QA?
Yes, we are considering this. So what we did in our case, we split the marker into 2. So we have the homozygous and then we have the heterozygosity. So the homozygosity markers for the 2 parents, we are sure about that one. But for the heterozygosity, this one could be on the error from the sequencing. So what we are doing for that one to avoid misleading a final conclusion, we sequence this one through the Illumina platform. So we complement this one with high density to avoid misleading information. But of course, we have to consider the error rate in the process.
Yeah, good to hear that. But in the case in which you use a few number of markers.
Yes. Like for the genetic purity, we use only 14 SNP markers to do this work. So we are using very few SNP markers. So from 99, we narrow it to 50, and then from 50, we narrow it down again to 14. So we are using very few SNP markers in the breeding program.
Okay, thank you.
Thanks.
So before we go to the next question of Gary is asking to Juan. Juan, Gary, do you want to ask to Juan directly, please?
Sure, Juan, that 20% to 30% error rate, is that on a per marker a per SNP basis or a per sample basis?
This per sample base.
Yes.
So 70-80% of the samples will be okay. 20 to 30% will have one or more miscalled SNPs.
Yes.
Is that right? Okay.
Yes.
That's right.
Okay, thanks. Yeah, that's not so bad.
So Gary, do you want to make your question?
Yeah, thanks very much for that very good presentation, Agre. That's great progress. I wonder how much validation you have to do on mid-density SNPs before you start, calculating the genomic relationship matrix and starting with cross prediction. What do you see as the validation process? We don't have to worry, I don't think, very much about individual SNPs, except in that they're spaced out well. There's good coverage, and they have a pretty good call rate, although that's probably not the most important factor, and that you have enough of them. What other factors are there?
Yeah. Thanks, Gary. What our approach first was kind of like when we select the 3,000 SNP markers, we are expecting at least during the call rate, probably we may end up with some marker that we fail during the conversion. So that's why we selected a lot of SNP markers. And then after the conversion, we are good. We have a very good coverage. So of course, we can now move directly now to generate the genetic relation matrix, which will be used for the cross prediction merit. So the reason behind selecting multiple markers is just to avoid, in case probably we have a lot of failure, we should not start going back and look for additional SNP markers. So that was the reason behind it.
That's great. So it sounds like you're ready to go on genomic prediction.
Yes.
Great.
So if anybody have any question, just raise the hand or unmute itself. Agre, I have a question, is more curiosity, because I know that identification of sex in YAM is extremely difficult. And as I understood, from one tuber, if we broke it, we can generate plants from different sex. Is this correct?
It may be really difficult because we don't have kind of like a very good information yet about that one. So I don't want to make the wrong conclusion. But the sex is still not yet very stable in YAM. It seems like it's still under evolution. But we have just to get more information about it before we could make a finally. Dr. Asrat also is on the call. I think he can provide the additional information if we can.
Asrat, you can unmute yourself, please.
Okay. Thank you. I think related with sex, the issue of sex switching and then express, non-express, it is only for the monoecious plant. But for the dioecious plant, the male continues to be male and the female continues to be a female. But that issue is there. But our marker can predict the male, okay, but the female, it's heterozygous. That means whenever you predict as a female, if it is monoecious, there is a chance to be turned into either male-dominated female monoecious with male-dominated flowers. So that kind of the problem is there. But our team colleague from the Japan, they are working more in depth into the areas and how that the sex determination also determine and then to come up with very good predictive the markers. So the work is going on.
Thank you. So another question is from Shayla. Shayla, do you want to unmute yourself and ask your question? Because Shayla is asking, why do you use these markers? However, we do not understand which markers you and for which trait you are asking.
Okay. We have different set of markers. We have the QC marker for routine activities in the breeding program. And then I don't really know if that one she's looking for. We have also the trait marker that we are intending to use for marker-assisted selection, especially at early generation to avoid the delay in selection process. So don't know if I answered very well her question.
Shayla, if you can interrupt anytime. Amy, do you want to say something about it?
Hello, can everybody hear me?
Yes, Amy.
Yes.
Yeah, so I am the regional manager for Africa at LGC in North Africa. So we do work on a lot of KASP projects, you know, ranging from one marker up to 192 markers. And then we have like a different product for higher marker range up to 30,000. And then we do whole genome projects. We do work with Summit at the moment, and if anyone else has any challenges, we're happy to work with different teams and to get some of our technical experts involved, if that's anything that that anyone in the call would ever like to reach out to us for.
Okay, thanks so much, Amy.
Maria, my colleague, is on the call and she's the application scientist, so she may have an input in this as well.
Thank you, Amy. Thank you very much.
You're very welcome, and it was very interesting. It was great to see all the research done. Thank you.
Thank you. Agre, there is a question from Mokhtar. Mokhtar, do you want to unmute yourself?
Oh yeah, thanks, Anna. I was just, and thanks, Agre, for your presentation. It was really nice to see the progress. I was wondering, how do you, what is your threshold? From what distance value do you say that two samples are different or are similar? And when you say that they are similar, how much confidence do you have in that information?
Oh, okay. So I get your point. So in the breeding program, we have done a kind of like a lot of work. So we come up with a threshold. So the genetic distance less than 0.03, which is kind of like 3% of the similarity. We can consider them as duplicates. So we are also working with Cornell to validate that threshold. So that is what we have for the YAM. So we have used several duplicate information, well-known duplicate genotypes, and then the genetic distance we have seen between those two samples is always less than 0.0333. So our threshold is from 0 to 0.03. That is our threshold. So based on that one, if the marker give us that confidence interval, that means we can really reliable on those marker.
Oh yeah, perfect. Thanks. It's even less than that what I was expecting. And thanks very much.
Okay, thank you.
Thank you. Not sure if there is more questions in the chat. There is no other questions. If there is no questions, I think we can come to the end of the second seminar of this series. And thank you very much. Thank you, Dr. Robert, Agre, Asrat, for all the support and for allowing us to know your work. Thank you very much to all the founders. And we hope to see you in the next seminar, that it will be on 7 September, where Dr. Sharifa Aoui will introduce Dr. Andekelik Moamoonge in his talk about markers and drought tolerance in sorghum. So work performed in Tanzania. Thank you very much also to Julie from the CGIAR Comms for all the support with the beautiful flyer. And thank you to all.
Okay, thanks so much, Anna.
Thank you.
Thank you.
Yes.
Thank you, Anna.
Thank you.
Yeah, thank you, Anna, and thank you all.
Thank you very
Thank
much.
you all.
Thank you.
Thank you.
Okay.
Bye.